So what do we do with that mRNA? Well, we take it for translation. And so once you have your English recipe, you take that and you put the ingredients together. All right, and this happens again in the ribosome. This is ribosomal area because now we're making the protein. It's going to involve those messenger RNAs, the transfer RNAs, and the ribosomal RNAs. And this is where you've got the triplet reading, three nucleotides at a time will represent a certain amino acid. And the tRNAs will bring in that amino acid and will bind to that codon sequence. So it's a 3-base sequence on the mRNA that the tRNAs will bring in an amino acid for. So for example, if the code on your mRNA was an AUU, or better yet, a CUC, then the tRNA that could bind that CUC would bring in the leucine amino acid. Okay? And so you can imagine there are a lot of codes here. You will not need to memorize those. I would give you on an exam the amino acid that's coded for by the codon sequence. So let's look in a little more detail at translation. Again, we have this mRNA that's being transcribed from the DNA template strand in the nucleus. It leaves the nucleus and heads to a ribosome. Most oftentimes it's going to happen on the ribosome of an ER, endoplasmic reticulum. So the mRNA then is going to bind to a ribosomal RNA or an rRNA. Remember, we talked about those 3 main types of RNA. This here, this red, is an rRNA. So it's going to bind at a start codon. And the anticodon is what we call the opposite of the 3 codon triplet here on the mRNA strand. So this is our mRNA strand. It's got 3 right here. The tRNA that is the complementary codon sequence of this will come in. So in this case we had CCG. The T with GGC will come in, and it will have the associated protein on it, or amino acid, excuse me. So this is proline. And so proline is GGC, and it's going to then say, hey, this is the next amino acid for this protein. So the anticodon of a tRNA binds to the complementary mRNA. And it keeps going along reading 3 by 3. This will continue to move in this direction, this mRNA. And so each codon is read, and a protein, the appropriate amino acid, is brought along and attached. So new amino acids are going to be added by these other tRNAs coming in. So the next one, if this is AUA, when it moves over here to the right, then UAU will come in and bring in its amino acid to be bound to this chain. And this is until you hit a stop codon, which is a sequence that will say, okay, we're done. That's your protein. And these amino acids, it's really important that you realize they are linked by peptide bonds, right? Because remember, we're making a polypeptide, so they are bound by peptide bonds. When we look at how the rough ER and protein synthesis is involved, it's obviously very important because it contains those ribosomes, and those ribosomes are sitting on the outside of the ER, and this allows kind of a place to catch a polypeptide that's being made or translated from this mRNA. And it can clean it up by getting rid of certain parts like the initial. It just cleans up the message overall, the protein overall, and can add sugar groups where needed so that they're off and ready to go to the next section, and they're transported in a vesicle. Let's look at development of cells. We've looked at a lot here in terms of cells divide, and all of our cells have the same DNA, every single one. This is why when there's a criminal investigation, You can get a blood sample from the wall. You can get a saliva sample, a semen sample. It doesn't matter. All of our cells have our DNA in them. They all have a complete readout of our DNA. But the cells themselves are not identical. And this is because they each have their own function. And so even though they all have the recipe, the complete DNA recipe in them, they're not going to necessarily express the same DNA. And so there are signals as we're developing in utero, as well as we're developing from children to adults. There are signals that are released at certain stages in our life, and they make stem cells go into a certain pathway, or convert into a certain developmental pathway. And this begins what we call differentiation of our cells. So some genes are expressed in red and make certain proteins, and other cells, depending on what their developmental pathway is, will express different genes or translate and make different proteins. And at the same time, we also have the elimination of cells that are not useful or have been damaged, or there's too many of them, or they're old. And so we do this through something called apoptosis or apoptosis. So depending on how you want to pronounce it. And this is known as programmed cell death.